Welcome to Chapter 24. We're going to talk about metabolism and nutrition. So the term metabolism, this refers to all of the chemical reactions that occur in the body. Now there are two types of metabolism, catabolism and anabolism. So we could actually write these in here. We will talk about them right away. Catabolism and anabolism. These are two types of metabolism. So the chemical reactions that break down complex organic molecules into simpler ones are known as catabolism. So overall, catabolic reactions are going to be exergonic. So again, these are going to be reactions that break down large, complex molecules into simpler ones. These are generally going to be exergonic. This means that they release heat as part of the reaction. Now, chemical reactions that combine simple molecules to form the body's complex structural and functional components, these are going to be anabolic reactions, or known as anabolism. So these are going to be taking simple molecules and building them or putting them together to build complex molecules. Now, an example of an anabolic reaction would be the formation of peptide bonds between amino acids during protein synthesis. Because we would take an amino acid and another amino acid, this whole bunch of amino acids, and in an anabolic reaction, we would bond those amino acids together to take the simple singular amino acids and bond them together into a larger, more complex molecule. So this would be anabolism. We can also think of catabolism as decomposition or breaking down, and anabolism as synthesis or creating things larger. Now, metabolism is an energy balancing act between the catabolic and anabolic reactions. Now, the molecule that participates most often in energy exchanges in living cells is going to be ATP, adenosine triphosphate. This is our energy molecule. So ATP couples energy releasing catabolic reactions to energy requiring anabolic reactions. So anabolic reactions, RxN is an abbreviation for the word reaction. So anabolic reactions, these require energy. And catabolic reactions, these release energy. So this picture is kind of how ATP releases energy, and it can kind of help anabolic reactions in the way that ATP can release the energy required by anabolic reactions. So here we have ATP. We kind of think of it as a full battery because it has this really high energy bond between the last two phosphate molecules. So this bond right here between these two phosphates is really high energy. And so when that bond breaks, so here we have this breaking of this bond, it releases that energy that was in the bond. So now we have this free energy. We have this free floating inorganic phosphate because it broke off from ATP, or that bond broke. And then the rest of this molecule is known as adenosine diphosphate, because now it only has two phosphate molecules attached, so di meaning two phosphates, and adenosine triphosphate, tri means three, so it has 1, 2, 3 phosphate molecules. So we go from this really high energy ATP to a very low energy ADP because it has released that free energy. And that energy can be used by other reactions in the area. Now, when complex molecules and polymers are split apart in catabolic reactions, some of that energy is transferred into ATP here. And then the rest of the energy that's released from this reaction is released as heat. Now, when simple molecules and monomers are combined to form complex molecules, this is anabolism. ATP is going to provide the energy for this synthesis. So it kind of gives its energy there. And then again, we have some energy released as heat. So this ATP to ADP reaction occurs at the same time that other molecules are going through this catabolism or anabolism, so that ATP can kind of provide the energy for them. Various catabolic reactions transfer energy into high energy phosphate bonds of ATP. Although the amount of energy in these bonds isn't exceptionally large, it can be released very quickly and very easily. Now, two important aspects of energy transfer are oxidation-reduction reactions and mechanisms of ATP generation. So oxidation is the removal of electrons from an atom or a molecule. Now, the result is a decrease in potential energy of the atom or molecule. One way we can remember oxidation and reduction reactions is the little helpful saying here, which is oil rig. OILRIG. It stands for oxidation is loss. This is of electrons. Remember here, so removal of electrons. Oxidation is loss. Reduction is gain. So that's going to be the gain of electrons that we'll see in the next slide, actually. So an example of an oxidation reaction is going to be this reaction right here at the bottom. This is the conversion of lactic acid into pyruvic acid. So in this reaction, hydrogen atoms are removed as one hydrogen atom and one hydroxide ion. So we remove two hydrogen ions, and there also is a removal of electrons that go with those hydrogen atoms. So this is an oxidation reaction. we're going to be losing 2 hydrogen atoms and losing electrons. Now, a reduction reaction is going to be the opposite of oxidation. So this is the addition of electrons to a molecule. Now, reduction results in an increase in the potential energy of the molecule. So we get an increase in the potential energy of the molecule, and an example of a reduction reaction is going to be kind of the opposite reaction that we just looked at on the last slide. So reduction reaction is going to be the conversion of pyruvic acid to lactic acid. So here we are adding 2 hydrogen atoms and adding the electrons that go with them. And so this will be a reduction reaction. Now, these oxidation and reduction reactions are important for our purposes because it has to do with metabolism and where we're going to be getting the energy required for certain reactions from. Now, when a substance is oxidized, the released hydrogen atoms don't remain free in the cell, but they're going to be immediately transferred by coenzymes to another compound. So coenzymes are going to be kind of our transferring molecules. Now, there are 2 coenzymes that are commonly used by animal cells to carry hydrogen atoms. These are nicotinamide adenine dinucleotide, also known as NAD. This is a derivative of B vitamin niacin. And the second one is flavin adenine dinucleotide, which is also known as FAD. This is a derivative of vitamin B2 known as riboflavin. So we usually just call these coenzymes NAD and FAD. Now, the oxidation and reduction states of NAD and FAD are represented in this little picture here. So when NAD is reduced to NADH and an hydrogen atom, this is where the NAD plus gains 2 hydrogen atoms. So the NAD is going to gain 1 hydrogen, which neutralizes its charge, gains this negative hydrogen atom. This is a hydride. Hydride ions are H negative. And so the NAD plus, because it has this positive charge associated with it, it will take the negative hydrogen and become a neutral atom or neutral molecule. And then the positive hydrogen ion is released into the surrounding solution. Going the other way, when NADH is oxidized into NAD+, there is a loss of the hydride ion from NADH. And this results in 1 less hydrogen atom and then the additional positive charge on the NAD. On the other hand, with FAD, when FAD is reduced to FADH2, it's going to gain a hydrogen atom and a hydride ion. And when FADH2 is oxidized to FAD, it's going to lose those same 2 ions. So that FAD is the oxidized version, and FADH2 is the reduced version. And then same with NAD being the oxidized version and NADH being the reduced version. These are our two coenzymes that will essentially carry hydrogen atoms to where they're needed. So now we're going to kind of put those two reactions together. So we saw what happens when one compound is going to be oxidized to another, or vice versa, reduced to the initial, like when we saw with lactic acid and pyruvic acid. And we also saw what happens with the coenzymes picking up or releasing hydrogen ions. Now this is important because oxidation and reduction reactions are always coupled. So that each time one substance is oxidized, there's going to be another one that is simultaneously reduced. For example, when lactic acid is oxidized to form pyruvic acid, where does it send its hydrogen atoms to? The two hydrogen atoms removed in this reaction are going to be used to reduce NAD into NADH. So this is how we have a coupled reaction. And this lactic acid pyruvic acid side of the coupled reaction here, this could really be any molecule, any oxidation reduction molecule. But it's always going to be coupled with a coenzyme like NAD or FAD. Now, an important point to remember about oxidation reduction reactions is that oxidation is usually exergonic, so it's going to be an energy releasing reaction. Now, some of the energy released during oxidation reactions is captured within a cell when ATP is formed. This is when a phosphate group, as is denoted as P, is added to ADP. It requires an input of energy to form ATP. Now, the two high-energy phosphate bonds that can be used to transfer energy within ATP are indicated by little squiggly lines. So these two here, these are going to be high-energy bonds. Now, the high-energy bond that attaches the third phosphate group to the rest of the group contains the energy. Pretty much contains most of the energy in this reaction. So this bond has the most energy to be able to be released within this molecule. Now, one other thing of note is that the addition of this phosphate group to a molecule, this is called phosphorylation. This is the addition of any phosphate group to any molecule, but this increases the potential energy. So phosphorylation increases potential energy. So anytime we add a phosphate group to a molecule, it requires energy to add it, but once that phosphate group is added to a molecule, there's an additional energy, potential energy that's stored within that molecule, and we can use that energy that's stored by that bond holding the phosphate on, and we can use that to power other reactions. So whenever we phosphorylate, whenever we add a phosphate group to a molecule, it increases the energy in that molecule. So it increases the amount of energy that can be released from that molecule as well. So as we learned in a prior chapter, both polysaccharides and disaccharides when they're hydrolyzed or when they're broken apart, they're going to be converted into the monosaccharides, glucose, fructose, and galactose. Oops. Galactose, that's a C there. So glucose actually makes up about 80% of the monosaccharides that are broken off from polysaccharides. And this is during the digestion of carbohydrates. But really, hepatocytes will actually convert the fructose and galactose into glucose. So these will actually be converted here by hepatocytes, those liver cells. So they're going to get converted into glucose anyway. So really, when we're talking about carbohydrate metabolism, it really is just glucose metabolism, because all of the monosaccharides are mostly going to be glucose anyway. So glucose is the body's preferred source for synthesizing ATP, and its use depends on the needs of body cells. Now, the needs of body cells include things like ATP production. So in body cells that require immediate energy, glucose is oxidized to produce ATP. The cells may also need amino acid synthesis. So cells throughout the body can use glucose to form several amino acids, which can then be incorporated into proteins. The cells may also need glycogen synthesis. So hepatocytes and muscle fibers can actually perform glycogenesis, this is where hundreds of glucose monomers are combined to form the polysaccharide glycogen. So glycogenesis is where glucose molecules are all kind of stacked and combined together to form this really, really large molecule called glycogen. Glycogen is going to be the stored form of glucose. It's kind of just putting like tons of boxes together, and then we call that stack of boxes by another name. Each box would be a glucose molecule, but then the stack of boxes is going to be glycogen. Kind of like that. And the cells may also need triglyceride synthesis. So when these glycogen stores are all filled up, there's no more space for any more glycogen, Then the hepatocytes can transform the glucose into glycerol and fatty acids that can be used for lipogenesis. Lipogenesis is the synthesis of triglycerides. So this is where we would take glucose and the hepatocytes would transform it into the glycerol and fatty acids to make triglycerides. Then those triglycerides are deposited in adipose tissue. That fatty tissue. Now, fatty tissue or adipose tissue has virtually unlimited storage capacity. So if you continue to intake carbohydrates and glucose after your glycogen stores are all filled, the hepatocytes will continue to make fat tissue or fill up that adipose tissue, continue to fill it with triglycerides, as long as you continue to intake excess glucose. Before glucose can be used by body cells, it must first pass through the plasma membrane to enter into the cytosol of the cell. Glucose absorption in the gastrointestinal tract and also in the kidney tubules is accomplished by secondary active transport. We saw that in the last chapter. Now, glucose entry into most other body cells occurs by these GLUT molecules, G-L-U-T. This is a family of transporter molecules that bring glucose into cells by facilitated diffusion. So these are all other cells of the body, not in the gastrointestinal tract, but all other body cells, glucose can enter using these GLUT molecules by facilitated diffusion. Now, a high level of insulin will actually increase the insertion of one type of GLUT molecule called GLUT4, and it will increase the insertion of these GLUT4 molecules into the plasma membrane of most body cells. So this will increase the rate of facilitated diffusion of glucose into cells. So if we increase insulin, this will increase the insertion of GLUT4 molecules. And this means we're going to increase glucose moving from the blood into the cells. And this is one way that insulin decreases blood glucose levels, because we're going to be quickly moving glucose from the blood into the cells. We're not getting rid of that glucose that's in the blood, but we're moving it into the cells so that it can be used by the cells. Now, once glucose is in the cells can start to use that glucose to produce ATP. So the oxidation of glucose to produce ATP is also known as cellular respiration. And cellular respiration involves 4 sets of reactions. These are glycolysis, the formation of acetyl coenzyme A, the Krebs cycle, and the electron transport chain, also known as the etc. So we will go over each of these individually. This is a more complex set of metabolic reactions, so it's going to be a lot to go over. So here we have a pictorial representation of the 4 phases of carbohydrate metabolism to break down glucose into ATP. So our first phase here is glycolysis. This is a set of reactions in which one glucose molecule is oxidized into 2 molecules of pyruvic acid. And during this set of reactions, there are 2 molecules of ATP that are created and 2 molecules of ADH, or NADH, sorry. Now these coenzymes, the NADH and FADH2, are going to be produced in the first 3 phases, but they're all only going to be started to be used during the 4th phase, the electron transport chain. Our second phase here is the formation of acetyl coenzyme A. This is really just a transition step that prepares pyruvic acid for entrance into the Krebs cycle. So pyruvic acid by itself can't actually go into the Krebs cycle. It has to be converted into acetyl coenzyme A. So each molecule of pyruvic acid is converted into an acetyl coenzyme A molecule. Now during this transition phase, there are also 2 molecules of carbon dioxide made and 2 NADH. Then we have the 3rd phase, which is the Krebs cycle. And these reactions will oxidize acetyl coenzyme A, and they will produce 2 molecules of ATP, 4 carbon dioxide, 6 NADH, and 2 FADH2. So then all of those coenzymes, NADH and the FADH2, are all going to be sent to the electron transport chain. So the electron transport chain, which is our 4th phase here, these reactions will oxidize NADH and FADH2 and transfer their electrons through a series of electron transfers. Now, the electron transport chain in the end produces 6 water molecules and 26 to 28 ATP molecules. So this is going to be the main phase where the most ATP is created. Now, glycolysis does not require oxygen. And so because of that, it can occur under aerobic, which is with oxygen, or anaerobic conditions. So anaerobic means without oxygen. So essentially, glycolysis can occur with or without oxygen, doesn't matter too much. In contrast, the Krebs cycle and the electron transport chain, these both require oxygen, and so collectively these 2 steps are referred to as aerobic respiration. So aerobic respiration just refers to the Krebs cycle and the electron transport chain. This intermediate step, this formation of acetyl-CoA, does require oxygen as well. So this can only occur under aerobic conditions too. But it's kind of not included in this overarching aerobic respiration term. That's only going to be the Krebs cycle and electron transport chain. So all that to say that if oxygen is present, all four phases will occur. However, if there is no oxygen available or very little oxygen, pyruvic acid will actually go down this anaerobic pathway, and it will be converted into a substance called lactic acid. And if pyruvic acid is converted into lactic acid under anaerobic conditions, then the remaining steps of cellular respiration cannot occur because we need this acetyl coenzyme A in order to finish the Krebs cycle and electron transport chain. So without oxygen, glucose will be converted into pyruvic acid and then lactic acid, and that's kind of where it stops. So oxygen is needed to to create the most amount of ATP from glucose. So first let's talk about glycolysis. During glycolysis, chemical reactions will split a 6-carbon glucose molecule into 2 3-carbon molecules of pyruvic acid. Now there are 10 steps in glycolysis, though you'll really just be required to know the main points of this part of cellular respiration. You don't have to memorize every step involved or anything like that. That would be far too complicated. So here on the right is our overview of glycolysis. Essentially, for our class and our purposes, this will be the extent to which you should know glycolysis. We start out with 1 glucose molecule, and it actually requires an input of 2 ATP molecules in order to kind of convert glucose to its first intermediate. You don't have to know what this is, but you do have to know that glucose will consume 2 ATP molecules, but in the end, it produces 4 ATP molecules. So what that means is that it produces 4, but we're going to consume 2 in the beginning. So we have a net 2 ATP molecules produced. So we have this net gain of 2 ATP molecules for each glucose molecule that's oxidized into 2 pyruvic acid molecules. So this figure here shows our 10 reactions that make up glycolysis. Again, like I said, you don't need to memorize all these 10 steps or anything like that. Pretty much you just have to know in the last slide that overview of glycolysis. But very briefly, I'm just going to go over this. So in the first half of the sequence, kind of steps 1 through 5 here, energy in the form of ATP is going to be invested. So we kind of have to put in some energy in form of ATP here and here in order to split up this glucose ring. Because splitting a glucose ring, or even any ring molecule, into chain molecules like we see over here, that takes some energy input. And so we use some energy in order to break it apart. One other possible important enzyme in this reaction is this enzyme here, phosphofructokinase. This is a really important enzyme in glycolysis, and it's a key regulator in the rate of glycolysis. So it's a key regulator in the rate of glycolysis, or how quickly glycolysis can proceed. Now, quickly with phosphofructokinase, Phosphofructokinase can have really high activity or can work very quickly when ADP is high. So then ATP is produced quite rapidly. So when ADP is in high concentration, phosphofructokinase is going to have high activity. But when the activity of phosphofructokinase is low, most glucose doesn't even enter the reaction of glycolysis. Instead, glucose is going to be converted into glycogen for storage. This is when phosphofructokinase activity is low. Now, in this second half of the sequence, kind of these steps 6 through 10 here, this is where we have 2 glyceraldehyde 3-phosphate molecules that are converted into 2 molecules of pyruvic acid. And it's going to be kind of the last half of glycolysis where we get the output of ATP. You can see here in step 7, we have 2 ATP molecules produced, and then in step 10, we have 2 molecules of ATP produced. So kind of just the takeaways of glycolysis is that it requires input of 2 ATP at the beginning. It has an output of 4 ATP to give us a net gain of 2 ATP. We also get 2 NADH produced. And then our last takeaway is that phosphofructokinase is a key regulator in the rate of glycolysis. Just meaning that if phosphofructokinase has low activity, then glycolysis can't really proceed very quickly, so it'll have a low rate or a low activity of phosphofructokinase will decrease the rate of glycolysis. And when there's a high activity of phosphofructokinase, then the rate of glycolysis increases. Moving on, the fate of pyruvic acid during glycolysis, or kind of following glycolysis, really depends on the availability of oxygen. This is really important. So if oxygen is really scarce, or there's no oxygen, this is going to be an anaerobic condition. For example, in skeletal muscle fibers during strenuous exercise, you kind of use up all of your oxygen during the first bit of exercise, and if you continue exercising, the amount of oxygen available to muscles decreases. So in that case, we would start to have an anaerobic condition. Then pyruvic acid is reduced by an anaerobic pathway into lactic acid. Now, when oxygen is really plentiful, as in aerobic conditions, most cells will convert pyruvic acid into acetyl coenzyme A. Now, this molecule, acetyl coenzyme A, is going to link glycolysis, which occurs in the cytosol, to the Krebs cycle, which occurs in the matrix of the mitochondria. So we'll actually see this in the next slide too, but just quickly. So glycolysis occurs in the cytosol, and the Krebs cycle, this occurs in the mitochondria. So pyruvic acid will enter into the mitochondrial matrix with the help of a special transporter protein. So each step in the oxidation of glucose usually requires a different enzyme and often a different coenzyme as well. For our purposes and for our class, we're going to define a coenzyme as a compound necessary for the functioning of another enzyme. So the coenzyme that's used in this point in cellular respiration is coenzyme A. We're going to see this down here. This is derived from a B vitamin. So this is kind of a transitional step between glycolysis and the Krebs cycle. And this is where pyruvic acid is prepared for entrance into the Krebs cycle. So up here in the cytosol, we see this glycolysis reaction converting glucose into 2 pyruvic acid molecules. And like we kind of just went over, if there is very little to no oxygen present, pyruvic acid will follow the anaerobic pathway here, being converted into lactic acid molecules. But if oxygen is present, it will follow the aerobic pathway, and pyruvic acid will be transported into the mitochondria by this special transport molecule. We don't really need to know what it is, but pyruvic acid is transported by the use of a transport molecule into the matrix of the mitochondria. So we can see pyruvic acid in here. It produces an NADH molecule, and a coenzyme A is added. It's a very kind of short transitional process, but really in the end we get that acetyl coenzyme A that can enter the Krebs cycle. So now it is ready to enter the Krebs cycle, whereas before, with just pyruvic acid, it was not able to enter into the Krebs cycle. But now this step has prepared this molecule to enter into the Krebs cycle. Now, once pyruvic acid has been converted into acetyl coenzyme A, now it is ready to enter into the Krebs cycle. The Krebs cycle, this is named for the biochemist Hans Krebs, who first described these reactions, but it's also known as the citric acid cycle. And this is because the first molecule formed when an acetyl group joins the cycle is citric acid. These reactions occur in the matrix of the mitochondria and consist of a series of oxidation and reduction reactions, as well as decarboxylation reactions that release carbon dioxide. In the Krebs cycle, the oxidation reduction reactions will transfer chemical energy in the form of electrons to 2 coenzymes. These are NAD and FAD. So the acetyl-CoA derivatives are going to be oxidized and the coenzymes are reduced. So this is how we get that oxidation reduction coupling. So the main takeaway from the Krebs cycle is pretty much this overview here. It's that acetyl-CoA will enter into the cycle. The cycle goes through one complete kind of turn of the cycle. And for each turn of the Krebs cycle, there are 3 NADH molecules created, 3 hydrogen ions, and 1 FADH2 molecule produced by the oxidation reduction reactions, and 1 molecule of ATP. So that's for 1 turn, 1 acetyl coenzyme A molecule entering into the cycle. Now, since each glucose molecule provides or creates 2 pyruvic acid molecules, so 1 glucose will create 2 pyruvic acid, and each pyruvic acid molecule will produce an acetyl coenzyme A molecule. So you get, for each glucose, you get 2 acetyl-CoA. So we're going to double these numbers. So these numbers are for 1 acetyl-CoA. For each acetyl-CoA that enters into the Krebs cycle and goes through 1 turn, these are the numbers that we get. So for each glucose molecule, we're going to double that. So this is the numbers for each glucose molecule that is oxidized, or for each, well, for 2 acetyl-CoAs. This figure shows the reactions of the Krebs cycle in more detail. This one is more just for your interest and information, because for this class we don't really need to memorize the Krebs cycle in detail or anything. We essentially just have to know the products of the Krebs cycle. Know the inputs and the outputs. So remember, we just saw this in the prior slide. So remember that each glucose molecule will produce 2 pyruvic acids, and thus 2 acetyl coenzyme A's. And then each acetyl CoA will produce 3 NADH for each cycle, 3 hydrogen ions, 1 FADH2, and also a molecule of ATP. The electron transport chain, this is a series of electron carriers which are integral membrane proteins in the inner mitochondrial membrane. So they will span the entire width of the phospholipid bilayer of the inner membrane. Now, the inner membrane of the mitochondria is folded up into cristae that increase the surface area and accommodate for thousands of copies of the transport chain in each mitochondria. So just a little quick picture here. If we look at a mitochondria, kind of looks like this, kind of kidney-ish shaped. It's a bit long on one side in either case. So it has this outer membrane, and then on the inside, it has an inner membrane, but it's like twisted up a lot. And so all these little twists are called cristae, and it goes the whole length of the whole thing. So this is going to be what the inner membrane looks like. So it has this really increased surface area, because if we were to unfold this whole thing, it would be really long. So the proteins, these electron carrier proteins, are all going to be on the inner membrane. So we can kind of see like little membrane proteins, sets of 4. So we can get tons of sets of 4 along this entire membrane. Huge increase in surface area, so we have a ton of these integral membrane proteins. So each carrier of the chain is going to be reduced as it picks up electrons and then oxidized as it gives up electrons. So the electrons will pass through the chain, and there's going to be a series of exergonic reactions that release small amounts of energy, and this energy is used to form ATP. Now in cellular respiration, the final electron acceptor of the chain is oxygen. Now, because this mechanism of ATP generation links chemical reactions with the pumping of hydrogen ions, this entire process is known as chemiosmosis. So it links these chemical reactions, which are the passage of electrons along the transport chain. Electrons will just abbreviate E negative. And it links that with the pumping of hydrogen ions. Now together, the chemiosmosis and the electron transport chain will constitute oxidative phosphorylation. So if we take chemiosmosis and together with the electron transport chain, these are going to both constitute oxidative phosphorylation. So this is how chemiosmosis works. We're going to start with our little passageway here, starting at number 1. So energy from NADH and hydrogen ions are going to pass along the electron transport chain here, and it's used to pump hydrogen ions from the matrix of the mitochondria into the space between the inner and outer mitochondrial membrane. So this is going to be this space out here. It's going to be a space between the inner and outer mitochondrial membranes. So we're kind of just looking at, we kind of see the square here, but this is looking at this membrane, the inner membrane. So there's going to be a membrane here. This is like membrane 2, and then we can kind of think of another membrane out here. This is the outer membrane. Outer membrane, and then here's the inner membrane, which is actually labeled right here. And then the outer membrane is going to be outside, and then this is going to be that space between the two membranes. So hopefully that kind of makes sense. So we're pumping hydrogen ions into the space between the membranes, this light blue part here. This is where we're going to have this buildup of hydrogen ions. Now, this mechanism of pumping hydrogen ions into this space between the membranes, this is going to be completed by proton pumps. And this is because hydrogen ions essentially consist of a single proton. Now, because we're constantly pumping these protons out into the space between the outer and inner membrane, we're really going to get this high concentration of hydrogen accumulating between the inner and outer mitochondrial membranes. So we get this really high concentration. So we are pumping hydrogen ions against their concentration gradient. So we really need that energy from NADH to help pump those hydrogen ions against their concentration gradient to really build up the concentration of hydrogen ions in this membrane here. So increase that concentration here. Now, because we have this huge concentration of hydrogen ions between the membranes, we can use that as energy. Because the hydrogen ions will naturally want to move down their concentration gradient, so we kind of use it like a waterfall, kind of how we use hydroelectric, or how we make power from hydroelectric dams. Water naturally wants to flow downhill, and so we use that to kind of activate motors, essentially. And so kind of this is similar in that way, where we can get ATP synthesis occurring as these hydrogen ions flow back into the mitochondrial matrix through these really special type of hydrogen channels in the inner membrane. So this is done by ATP synthase. It kind of works as a motor that's powered by hydrogen ions flowing down their concentration gradient through this protein. So as you can see here, they will flow down, they'll go through this little mechanism and down back into the inner matrix of the mitochondria, but they're flowing down their concentration gradient. And so ATP synthase kind of acts like a motor where hydrogen is kind of its fuel. Check out this YouTube channel or this YouTube video here to kind of see how this ATP synthase works. So in your notes, you can copy and paste that and kind of see the ATP synthase in action. So several types of molecules and atoms will serve as electron carriers. Things like flavin mononucleotide, FMN, cytochromes. These are proteins with an iron-containing group. We know it as a heme. The electron transport chain actually contains many cytochrome carriers. So these are going to be proteins with a heme. Remember, hemes are going to be iron-containing groups. There's also iron-sulfur centers, and they will contain either 2 or 4 iron atoms. Copper atoms also act as electron carriers, and also coenzyme Q. Often we just call this Q. This is actually a non-protein carrier that is mobile in the lipid bilayer of the inner membrane, and we'll see it a little bit later. Now, most of these electron carriers actually combine in different combinations to create the main proton pumps in the electron transport chain. Now, the most important carrier proteins that are standalone carrier proteins in our electron transport chain are cytochrome C and coenzyme Q. We also just call this one Q. Within the inner mitochondrial membrane, the carriers of the electron transport chain are clustered into 3 complexes, each of which acts as a proton pump that expels hydrogen ions from the mitochondrial matrix and helps to create an electrochemical gradient of hydrogen. So they create this hydrogen electrochemical gradient. So in other words, each of the 3 proton pumps transports electrons and also pumps hydrogen into the space between the outer and inner mitochondrial membrane. So here we have the full electron transport chain, so you can see a whole pictorial representation of it. You can also check out this YouTube channel at the bottom here to kind of see how it all works, but we will go over it all. So the pumping of hydrogen ions produces both a concentration gradient of protons and an electrical gradient. So the buildup of these hydrogen ions makes one side of the inner mitochondrial membrane positively charged. So we can see these positive charges here, compared with the other side, which is going to be more negatively charged. So we get this resulting electrochemical gradient that has potential energy. And this potential energy created by the electrochemical gradient is called the proton positive force. So proton channels in the inner mitochondrial membrane, specifically ATP synthase, will allow hydrogen to flow back across the membrane, and this is driven by that proton positive force. So let's talk specifically about each of these pumps. So each of these pumps is a complex of 3 or more electron carriers. So our first pump, we're going to start on the left here. It's one labeled number 1. This is the first proton pump, and it's called NADH dehydrogenase complex. This is the name of this pump. And it contains FMN and 5 of these iron sulfur centers. So those are the electron carriers all within this proton pump. At this pump, NADH is oxidized to NAD+, and the electron carriers in the NADH dehydrogenase complex are going to be reduced, passing the electrons along. So these electrons are here. So these electrons get passed on to Q. So we can follow these electrons here. And these hydrogen ions, these will get passed up into the space between the outer and inner mitochondrial membranes. So kind of 2 things happen at each of these proton pumps, or at least at this one we'll start off with. The electrons from NADH are passed on to the next carrier, And at the same time, hydrogen ions are pumped out to the other side of the membrane. So Q, this is a mobile unit in the membrane. And so it's going to kind of move back and forth in the membrane. So we can kind of imagine it's going to move this way, and it's going to shuttle electrons to the second pump complex. So we'll take these electrons and move them to the next one. So we can kind of see that with this arrow here. And then we get to the second proton pump, labeled number 2 here. This is the cytochrome BC1 complex. Now this contains cytochromes, so cytochrome B1 and cytochrome C1, and it also contains an iron sulfur center. So electrons are passed through these electron carriers within the complex. So you can see these electrons will pass through, and at the same time that these electrons are passing through the cytochrome BC1 complex, it's also going to be taking these free hydrogen ions and pumping them out into the space between the two membranes. Now the next complex that's going to take the electrons is cytochrome C. So its electrons are going to go to cytochrome C, which is also a mobile shuttle. And it's going to pass the electrons from the second pump complex to the third pump complex. Electrons go to cytochrome C, and then they pass on to the third pump complex. So now the third proton pump, labeled number 3 here, is the cytochrome oxidase complex. Now this contains some cytochromes, cytochrome A and cytochrome A3, and it also contains 2 copper atoms. Now these electrons are passed through these electron carriers, and then it will pass its electrons to 1/2 of a molecule of oxygen. So we can kind of see this 1/2 of a molecule of oxygen. So electrons will go to that. The oxygen will become negatively charged, and it will pick up 2 hydrogen atoms from the surrounding medium to form water. So those electrons are required for this reaction. Now each of these complexes moves hydrogen atoms into the space between the inner and outer mitochondrial membranes, and it creates this really big electrochemical gradient with a huge amount of potential energy. So this space here, because we have so many hydrogen ions and this huge electrochemical gradient, we have this massive amount of potential energy. Now, as hydrogen ions flow back, because they want to go back, they want to travel down their concentration gradient, these hydrogen ions will actually generate ATP because hydrogen channels will also include the enzyme ATP synthase. Now this enzyme uses that proton positive force or proton motive force to synthesize ATP from ADP and a phosphate molecule. Now the process of chemiosmosis is responsible for most of the ATP produced during cellular respiration. So again, just briefly how ATP synthase works is that these hydrogen ions will flow or want to naturally flow down their electrochemical gradient, and they'll go through this hydrogen channel in ATP synthase. And as it travels through, ATP synthase kind of acts like a motor to use that energy from hydrogen moving down their concentration gradient to create ATP from ADP and a phosphate ion. The various electron transfers in the electron transport chain will generate either 26 or 28 molecules of ATP for each molecule of glucose that's catabolized. So that's from the electron transport chain. Now, the specifics of this you don't really need to know, but in brief, this discrepancy in ATP numbers produced is due to the differences in NADH shuttles. So whether we're going to get 26 or 28 just depends on differences in NADH shuttles in different areas of the body. That's kind of the gist of it. So kind of like because of this, the cells of the liver, kidneys, and heart will actually produce an average of 2 more ATP per glucose molecule than cells in other parts of the body. So for example, cells of like the skeletal muscle fibers or neurons are going to produce 26 ATP. Liver, kidneys, heart are going to have 28 ATP produced, and these are just because of different NADH shuttles. So now recall that we have 4 molecules that are produced, 4 molecules of ATP produced prior to the electron transport chain. So we have 2 molecules of ATP from glycolysis. It's going to be 2 ATP there, and then also from the Krebs cycle, we're also going to get 2 ATP from there. And so if we kind of add all those up, we're going to get a total of either 30 or 32 ATPs produced from 30 to 32 ADPs. And this is from each molecule of glucose starting out. So this overall reaction where we have 1 molecule of glucose, 6 oxygen molecules, and 30 or 32 ADPs, and also 30 or 32 phosphate molecules, will produce 6 carbon dioxide molecules, 6 water molecules, and then 30 or 32 ATP molecules. So quite a lot of ATP produced from all of cellular respiration. This figure here shows kind of a summary of the principal reactions of cellular respiration. So at the top, occurring in the cytosol, we have glycolysis, converting 1 molecule of glucose into 2 pyruvic acid. The pyruvic acid is transported into the mitochondrial matrix, where it is converted into acetyl coenzyme A, which enters the Krebs cycle. Then we have the coenzymes that are produced during the Krebs cycle, NADH and FADH2, are sent to the electron transport chain down here, where most of the ATP is going to be produced. So this is a really nice summary table that summarizes the amount of ATP that is yielded during cellular respiration. Very, very helpful here. Even though most of the glucose in the body is catabolized to generate ATP, glucose may take part in or be formed by several anabolic reactions. So if glucose is not needed immediately for ATP production, it combines with many other molecules of glucose to form glycogen. Glycogen is a polysaccharide that is the only stored carbohydrate in the body. The hormone insulin from pancreatic beta cells will actually stimulate hepatocytes and skeletal muscle cells to carry out glycogenesis. This is going to be the synthesis of glycogen from glucose molecules. So this is stimulated by insulin, which stimulates those pancreatic beta cells. When body activities do require ATP, the glycogen that's stored in hepatocytes is broken down into glucose and released into the blood to be transported to cells, where it is going to be catabolized by the process of cellular respiration that we've already gone over. Now, this process of splitting down glycogen into its glucose subunits is called glycogenolysis. So don't confuse glycogenolysis, which is the breakdown of glycogen into glucose, with glycolysis, which is those 10 reactions that convert glucose into pyruvic acid. So when we're talking about glycogenolysis, this is going to be glycogen being broken down into glucose so that it can be used for metabolism. Glycogenolysis is not a simple reversal of the steps of glycogenesis. So on the left side here with the blue arrows, it shows glycogenolysis. Now remember, glycogenolysis is going to be that breakdown of glycogen to glucose. And then on the right side with the red arrows is the pathway of glycogenesis. And this is going to be creating glycogen from glucose. So they are a little bit different, just understanding that they're not completely just the simple reversal, but they do have a little bit different steps. The glycerol part of triglycerides, lactic acid, and even certain amino acids can be converted in the liver to form glucose. Now, the process by which glucose is formed from these non-carbohydrate sources is called gluconeogenesis. Now, gluconeogenesis is stimulated by cortisol, which is the main glucocorticoid hormone of the adrenal cortex. It's also stimulated by glucagon from the pancreas. And even the thyroid hormones, T3 and T4, these will also mobilize proteins and may mobilize triglycerides from adipose tissue, thereby making glycerol available for gluconeogenesis. So we have gluconeogenesis here in this diagram. As you can see, we have glycerol, lactic acid, and even some amino acids, and also pyruvic acid. that can be converted to make glucose. Now, an easy way to distinguish this term, gluconeogenesis, from other terms like glycogenesis or glycogenolysis is to remember that in this case, glucose is not converted back from glycogen. Instead, it is newly formed. So this neo part of the gluconeogenesis is like a new glucose. And so we get this new glucose from these other non-carbohydrate substances. So let's move on to lipid metabolism. Now, most lipids, like triglycerides, are going to be nonpolar, and so they're going to be very hydrophobic molecules. So they don't dissolve in water, and in order to be transported in the very watery blood, these molecules must first be made more water soluble by combining them with proteins produced by the liver and intestine. The lipid and protein combinations that are formed are called lipoproteins. Kind of makes sense, those two words together, lipids and proteins. So these are called lipoproteins. Now these are spherical particles with an outer shell of hydrophilic proteins and phospholipids and cholesterol that surround an inner core of triglycerides and other lipids that are very hydrophobic. So here we have a lipoprotein. You can see we have this single layer of amphipathic phospholipids. So we have like amphipathic phospholipids with their hydrophilic head towards the outside and their hydrophobic tails towards the interior nonpolar lipids. So inside we have some cholesterol, triglycerides, some very nonpolar, very hydrophobic substances on the inside, and then some cholesterol and additional proteins, these big proteins on the outside, surrounding this nonpolar lipids on the inside. Now, there are two sources of cholesterol in the body. Now, some of it is present in foods, things like eggs, dairy products, organ meats, beef, pork, but most of our cholesterol is synthesized by hepatocytes, those liver cells. So as total cholesterol level increases, the risk of coronary artery disease also begins to rise. Among the therapies used to reduce blood cholesterol, we have exercise, diet, and certain drugs. Regular physical activity at aerobic or nearly aerobic levels will rise HDL levels. Dietary changes are aimed at reducing the intake of total fats, saturated fats, and cholesterol. And then some of the drugs used to treat high blood cholesterol will promote the excretion of bile in the feces. So let's talk a little bit specifically about lipid metabolism. So just like carbohydrates, lipids may also be oxidized to produce ATP. If the body has no immediate use or immediate need to use lipids in this way, they're going to be stored in adipose tissue or fat deposits throughout the body and even in the liver. A few lipids are used as structural materials or used to synthesize other essential substances. So some examples of these structural materials would be things like phospholipids in the cell membranes, lipoproteins, and even the myelin sheaths around neurons are also where we use lipids. A major function of adipose tissue is to remove triglycerides from chylomicrons and VLDLs and to store them until they're needed for ATP production in other parts of the body. Triglycerides that are stored in adipose tissue actually constitute 98% of all body energy reserves. So a lot of energy is stored in adipose tissue. They're actually stored more readily than glycogen, and this is partly because triglycerides are hydrophobic, and so they don't exert osmotic pressure on the cell membranes. In order for muscle, liver, and adipose tissue to oxidize the fatty acids derived from triglycerides to produce ATP, the triglycerides must first be split into glycerol and fatty acids. So this is a process called lipolysis. This is lipid catabolism. So again, this is completed mainly by muscle, liver, and adipose tissue. These are going to be the tissue types that can perform lipolysis. Liver cells and adipose cells can synthesize lipids from glucose or amino acids. And this is through lipogenesis. So remember, genesis meaning new or beginning. So we're going to be creating new lipids. Now this is a process that is stimulated by insulin. Lipogenesis occurs when individuals consume more calories than are needed to satisfy their ATP needs. So this is lipid anabolism. And again, this is completed by liver cells and adipose cells. Now here are the pathways of lipid metabolism. So you can see that triglycerides here must first be split into glycerol and fatty acids, the process being lipolysis as the green arrows. So we can see that breaking up triglycerides into glycerol and fatty acids is going to be these green arrows here denoting lipolysis. And then glycerol may be converted into glyceraldehyde 3-phosphate, which is one of the intermediates of glycolysis, which can then either be converted into glucose or pyruvic acid. If it's converted into pyruvic acid, it can then be turned into acetyl-CoA to go to the Krebs cycle for oxidation. And fatty acids will actually undergo a process known as beta oxidation to enter into the Krebs cycle via acetyl-CoA. Now, the orange arrows are going to be lipogenesis. Again, this is stimulated by insulin. And this is the synthesis of lipids from glucose or amino acids. So you can kind of follow these arrows from amino acids all the way to triglycerides here. And then as well from glyceraldehyde 3-phosphate, taking the glycerol route here. Now let's move on to protein metabolism. So during digestion, proteins are going to be broken down into amino acids. Now, unlike carbohydrates and triglycerides, which are stored, proteins are kind of not warehoused, or they're not stored for future use. Instead, amino acids are either oxidized to produce ATP, or they're going to be used to synthesize new proteins for body growth and repair. Almost immediately after digestion, amino acids are reassembled into proteins. Many proteins function as enzymes. Others are involved in transportation, like hemoglobin would be an example of a transportation protein. Hemoglobin's example, transporting oxygen and carbon dioxide in the blood. Some will serve as antibodies. Clotting chemicals would be another type of protein. I'll do example here. An example of a clotting protein would be something like fibrinogen, like we saw with the clotting factors. Some proteins would be hormones, something like insulin. Or even the contractile elements in muscle fibers. So think of an example like actin and myosin. These are all going to be different types of proteins with very different functions. This table here just gives a list of different types of proteins and their functions. from structural to regulatory, even immunological. So lots of stuff we've talked about in the past throughout the whole class. So just a nice summarizing table here has everything in one place. A certain amount of protein catabolism occurs in the body each day. This is generally stimulated by cortisol from the adrenal cortex. Some of these amino acids are converted into other amino acids. And hepatocytes will convert some amino acids into fatty acids, ketone bodies, or even glucose. Now, cells throughout the body will oxidize a small amount of amino acids to generate ATP via the Krebs cycle and the electron transport chain. Now, protein anabolism, remember this is the formation of new peptide bonds between amino acids to produce new proteins, is carried out on the ribosomes of almost every cell in the body. And this is directed by the cell's DNA and RNA. Now this is a big figure you don't necessarily have to memorize or anything, but this is just to show that before amino acids can be oxidized to produce ATP, they first have to be converted into molecules that are part of the Krebs cycle or molecules that can enter the Krebs cycle, like acetyl-CoA. So in these little yellow boxes, we have the 20 amino acids that we have in our body. Things like phenylalanine can be converted into tyrosine, which is another amino acid, and tyrosine can be converted into fumaric acid, which is an intermediate of the Krebs cycle, so it can enter in that way. Other things like glutamic acid can be converted into alpha-ketogluteric acid, which again is another intermediate in the Krebs cycle. A lot of these amino acids at the top of this figure can all be converted into acetyl-CoA with 1 or 2 steps. And so acetyl-CoA, obviously, we know can enter the Krebs cycle. And so this is all just to say that essentially all the amino acids can't enter into the Krebs cycle and be oxidized to produce ATP right away. They first have to be converted into molecules that are part of the Krebs cycle, that are an intermediate in the Krebs cycle already, or they have to be converted into molecules that can enter the Krebs cycle, like acetyl-CoA. So remember that this picture on the right is how proteins are made. So this is called protein anabolism. This is how proteins are made at the ribosome. It's the formation of peptide bonds between amino acids to create proteins. So remember here we have the small subunit and the large subunit of the ribosome that come together on the mRNA molecule that codes for a protein. And then we have our tRNA molecules that will bring the amino acids with them to the ribosome. And they will bring the correct amino acid according to the code set in the mRNA. They come close together in the ribosome, and then new peptide bonds are formed between those amino acids. until we get kind of this chain going. Even though there are thousands of different chemicals in cells, there are 3 molecules that play really pivotal roles in metabolism. So these are glucose 6-phosphate, pyruvic acid, and acetyl coenzyme A. So we kind of say that these molecules stand at metabolic crossroads. So these are really, really important molecules. Also, as I'll talk about a little bit shortly, the reactions that occur or don't occur, these metabolic reactions, really also depend on the nutritional activity status of the individual. So first off, we'll look at glucose 6-phosphate. So quite shortly after a glucose molecule enters a cell, there's going to be an enzyme that will actually quite quickly convert it to glucose 6-phosphate. Now there are 4 possible fates that await glucose 6-phosphate. The first one would be the synthesis of glycogen. So when glucose is really, really abundant in the bloodstream, like after you have a large meal, there's a large amount of glucose 6-phosphate that is used to synthesize glycogen. So we can store that glucose form in glycogen. The next fate that glucose 6-phosphate may go through is the release of glucose into the bloodstream. So if the enzyme glucose 6-phosphatase is present and active, glucose 6-phosphate can be dephosphorylated into glucose. So glucose 6-phosphate really just means that we add a phosphate group on the 6th carbon. And so if this enzyme, which is called glucose 6-phosphatase, this will actually convert glucose 6-phosphate back into glucose to be released back into the bloodstream to increase our blood glucose level. Anything essentially that ends in ase is going to be an enzyme. Our next fate of glucose 6-phosphate is the synthesis of nucleic acids. So glucose 6-phosphate is actually a precursor used by cells throughout the body to make ribose 6-phosphate, which is a 5-carbon sugar that's needed for the synthesis of RNA and DNA. So in this case, we would take glucose 6-phosphate and we would convert it into ribose 6-phosphate, which can then be used to make DNA and RNA. Oops. And then our last fate that glucose 6-phosphate can go through is glycolysis. So some ATP we know is produced anaerobically by glycolysis, and this is where glucose 6-phosphate is converted into pyruvic acid, which is another key molecule in metabolism. But if it goes through glycolysis, there's some ATP that's created, about 2 ATP. Each 6-carbon molecule of glucose that undergoes glycolysis yields 2 of these 3-carbon molecules of pyruvic acid. We know that because we went over glycolysis earlier. Now, this molecule, just like glucose 6-phosphate, stands at a metabolic crossroads. So it's kind of like we kind of build this crossroads here. So it could go one way or the other way. So if there's enough oxygen, then the aerobic reactions of cellular respiration can proceed. So let me go through. So we go through aerobic cellular respiration. If there's no oxygen or very little oxygen present, then we would have anaerobic reactions occur. So these are kind of the crossroads I'm kind of talking about, these metabolic crossroads here. So a couple different things that can happen to pyruvic acid. If that oxygen is in short supply, and we kind of take this right route that I've drawn out here and we go through anabolic reactions, this is when we would get that production of lactic acid. So if oxygen's in short supply, as in actively contracting skeletal or cardiac muscle, remember some of that pyruvic acid is going to be converted into lactic acid. Pyruvic acid can also be converted into alanine. So kind of in this way, carbohydrate and protein metabolism, because alanine is an amino acid, they're kind of, so carbohydrate and protein metabolism are linked by pyruvic acid in this way, because pyruvic acid can be converted into alanine, which is an amino acid used in protein metabolism. One other thing that can happen is gluconeogenesis. So pyruvic acid and certain amino acids can be converted into oxaloacetic acid, which is one of the Krebs cycle intermediates, which then in turn can be used to form glucose 6-phosphate. So remember, gluconeogenesis is going to be the production of glucose from non-carbohydrate sources. So pyruvic acid and then some amino acids. So amino acids are going to be non-carbohydrate sources. Some of these can be converted into oxaloacetic acid. This is a Krebs cycle intermediate. And then this oxaloacetic acid can be used to turn into glucose 6-phosphate, which we know can be converted, glucose 6-phosphate, we know can be converted back into glucose as well. Just as we learned actually in the previous slide, where we saw all the things that can happen with glucose 6-phosphate. When ATP level in a cell is low, but oxygen is really plentiful, most pyruvic acid is going to stream towards these ATP producing reactions. So these are the Krebs cycle and the electron transport chain, and it's going to do so by converting into acetyl coenzyme A. So this is when there's low ATP and high oxygen levels. And then pyruvic acid will be converted into acetyl-CoA. Now, acetyl-CoA is going to be involved in the entry into the Krebs cycle. So remember that acetyl-CoA is the vehicle for 2 carbon acetyl groups to enter into the Krebs cycle. It's going to be kind of that intermediate between pyruvic acid and going into the Krebs cycle, so it's very important there. And acetyl-CoA is also involved in the synthesis of lipids. So acetyl-CoA can be used for the synthesis of certain lipids. These include fatty acids, ketone bodies, and cholesterol. Here we have the summary of the roles of the key molecules in metabolic pathways. Now, double-headed arrows, like the one we see here, these are going to indicate that reactions between the 2 molecules on either side of these arrows, they may proceed in either direction. So that is if the correct enzymes are present and also if conditions are favorable. So just a quick note on that. Okay, so first we'll look here at glucose 6-phosphate up on the top left. So number 1, 2, 3, and 4 are involved in the fate of glucose 6-phosphate. Number 1 being the synthesis of glycogen here. And so the synthesis and breakdown of glycogen occur mainly in the skeletal muscle fibers and hepatocytes in the liver. Number 2 here is the release of glucose into the bloodstream. Again, these can happen either way, both of these actually that we talked about. So once glucose is released from the phosphate group, from glucose 6-phosphate, it can leave the cell and enter back into the bloodstream. Hepatocytes are going to be the main cells that can provide glucose into the bloodstream this way. Number 3 here is the synthesis of nucleic acids. So remember that glucose 6-phosphate is used to make ribose 5-phosphate. I might have said ribose 6-phosphate before, but ribose 5-phosphate is what is made from glucose 6-phosphate. This is that 5-carbon sugar that's used to make DNA and RNA. And our number 4 here is glycolysis. So actually most body cells are able to carry out glycolysis, and some ATP is produced when glucose 6-phosphate is converted into pyruvic acid. Now, numbers 5, 6, and 7 are going to be involved in the fates of pyruvic acid. So we'll cover these. Our number 5 one first is going to be the production of lactic acid. So this occurs by anaerobic conditions. Remember, this is with no oxygen or without oxygen. Then pyruvic acid is converted into lactic acid. Pyruvic acid in number 6 here can also be converted into alanine, remember, which is an amino acid. And then we have number 7. And number 7 here, it's not specifically labeled, but number 7 is actually gluconeogenesis. And so this is where pyruvic acid, as well as certain amino acids, can be converted into oxaloacetic acid. which is one of the Krebs cycle intermediates. And this in turn can be used to form glucose 6-phosphate up here. And it can also be used to go to the electron transport chain. Not the glucose 6-phosphate. The oxaloacetic acid goes through the Krebs cycle and then sends the NADH and FADH2 to the electron transport chain. And then lastly, we have reactions 8 to 10 are going to be involved in the role of acetyl coenzyme A. So first with number 8 here, if ATP is low, but oxygen is really plentiful, then through aerobic reactions, then pyruvic acid is converted into acetyl-CoA. Acetyl-CoA at number 9 can also enter into the Krebs cycle, and it will enter into the Krebs cycle to create those reduced coenzymes, NADH and FADH2, to create ATP through the electron transport chain. Then lastly, acetyl coenzyme A can be used, number 10 here, in the synthesis of lipids. So it can be used to create certain lipids, including fatty acids, cholesterol, and ketone bodies. So essentially this is the big summary picture of the key molecules at metabolic crossroads. Those 3 major molecules: glucose 6-phosphate, pyruvic acid, and acetyl coenzyme A. So this table is really great because it summarizes the different types of metabolism that we've talked about, and it helps you to kind of categorize them so that you can remember them better. So we have our carbohydrate metabolism here, lipid metabolism, and protein metabolism there. Now, regulation of metabolic reactions depends both on the chemical environment within body cells, such as the level of ATP and oxygen, and it also depends on signals from the nervous and endocrine systems. Some aspects of metabolism do depend on how much time has passed since the last meal. This will get us talking about the absorptive state. So during this absorptive state, the ingested nutrients are entering the bloodstream, and glucose is really readily available for ATP production. Soon after a meal, and the food is in your gastrointestinal tract, the nutrients will then start to enter the blood. Now remember that in the blood, the digested food that reaches this bloodstream is mainly going to be glucose, amino acids, and triglycerides in chylomicrons. So here we have a diagram showing the reactions that dominate the preabsorptive state. So first we have our number 1 in the top right corner here. About 50% of the glucose that's absorbed from a typical meal is going to be oxidized by most tissue cells. These are going to be cells throughout the body. And this oxidation of glucose is going to produce a lot of ATP by glycolysis, the Krebs cycle, and the electron transport chain. Remember that there'll be a little bit of water and CO2 made with that oxidation as well. Okay, moving on to number 2 here. Here we have the hepatocytes in the liver, and most glucose that enters into the hepatocytes is going to be converted into glycogen. There are also going to be small amounts that may be used for the synthesis of fatty acids and glyceraldehyde 3-phosphate, and these are generally converted into triglycerides in the liver. Now, some of the fatty acids and triglycerides that are synthesized in the liver will stay there, but the hepatocytes will actually package most of these triglycerides into VLDLs, which will carry lipids as triglycerides into adipose tissue for storage down here. Now, adipocytes will also take up some glucose that is not picked up by the liver. So this will be for number 4 here. And they're also going to convert it into fatty acids and glyceraldehyde 3-phosphate into triglycerides to be stored as well. There also is some glucose that is taken up by skeletal muscle, and it's going to be converted into glycogen for storage there. Okay, moving on to number 5 here. So number 5 we have down here. Now, most dietary lipids, mainly triglycerides and fatty acids, are going to be stored in adipose tissue. Only a small portion is going to be used for synthesis reactions. So remember that adipocytes obtain the lipids from the chylomicrons and from the VLDLs, as well as some of their own synthesis reactions. Many absorbed amino acids, these are going to enter the hepatocytes in the liver, and they're going to be deaminated into keto acids. Now, these keto acids can either enter the Krebs cycle for ATP production, or they can be used to synthesize glucose or fatty acids. So that's with amino acids. Now, some amino acids that enter hepatocytes are also going to be used to synthesize proteins. For example, plasma proteins of cells are going to be made from these ingested amino acids. Lastly, amino acids that are not taken up by hepatocytes are going to be used in other body cells, like muscle cells, for the synthesis of proteins or even regulatory chemicals like hormones or enzymes. Then, approximately 4 hours after the last meal, the absorption from the small intestine is going to be nearly complete, and blood glucose levels start to fall. because glucose continues to leave the bloodstream and enter body cells while no more is being absorbed from the gastrointestinal tract. Right? So at this point, blood glucose levels are starting to drop. Because we're continually reabsorbing glucose from the blood into our body cells, but we haven't eaten anymore, so there's no more glucose coming back into the blood from the gastrointestinal tract. So this is where we get our post-absorptive state. So during the post-absorptive state, both glucose production and glucose conservation help to maintain a healthy blood glucose level. Hepatocytes produce glucose molecules and export them into the blood. And other body cells will switch from glucose to alternative fuels for ATP production in order to conserve glucose, which is starting to be very scarce. This diagram here shows the major reactions of the post-absorptive state that produce glucose. Are going to be from 1 to 9 here. So again, we'll just start with number 1 here. So this is going to be the breakdown of liver glycogen. So essentially 4 hours after your last meal, you're kind of starting into a fasting state, or your body considers it a fasting state. Now, during fasting, a major source of blood glucose is going to be liver glycogen, which can provide about a 4-hour supply of glucose. So liver glycogen is going to be continually being formed and broken down as needed. At number 2 here on the top, we have lipolysis. So remember, lipolysis is going to be the breakdown of triglycerides into glycerol and fatty acids. Glycerol is also going to be used to form glucose in the blood. Number 3, just at the bottom of the liver here, this is gluconeogenesis using lactic acid. So that's what this one is. So during exercise, skeletal muscle tissue will break down stored glycogen and produce some ATP anaerobically by glycolysis, where we get this lactic acid from. Number 4 is also going to be gluconeogenesis, but it's going to be using amino acids. So we're going to get gluconeogenesis using amino acids in the skeletal muscle tissue, in the liver, and also in other tissues here. So we kind of get small amounts of breakdown of proteins in skeletal muscle and other tissues that releases large amounts of amino acids, which then can be converted to glucose in the liver by gluconeogenesis. So they kind of break down their proteins in other tissues and muscle, send it through the blood to the liver, where the liver the hepatocytes in the liver are going to convert those amino acids into glucose. Next, we have the oxidation of fatty acids as our number 5. So the fatty acids released by the lipolysis of triglycerides can't really be used for glucose production because acetyl-CoA can't be readily converted back to pyruvic acid. But most cells can actually oxidize fatty acids directly. So it'll feed them into the Krebs cycle as acetyl-CoA and then produce ATP through the electron transport chain. So get that in most other tissues, in the heart, and even in skeletal muscle. Then, number 6 is going to be our oxidation of lactic acid. So cardiac muscle, on this top right for the heart, cardiac muscle can actually produce ATP aerobically from lactic acid. It's a great way to produce ATP when there's not much glucose present. At number 7 will be the oxidation of amino acids. So just happens in the liver in the hepatocytes. Amino acids can be oxidized directly to produce ATP. Then we have our number 8, which is the oxidation of ketone bodies. So hepatocytes up in the liver, these can also convert fatty acids to ketone bodies. And then those ketone bodies are sent to the blood. And ketone bodies can actually be used by the heart, the kidneys and other tissues, as well as the brain to produce ATP. Now, one interesting thing is that the brain really only likes to use glucose for ATP production. The brain does not really use much else, but in kind of extreme starvation mode, the brain will finally use ketone bodies, kind of if there's no other way. But that's going to be the only way that the nervous tissue, the brain, will use ketone bodies. The heart and kidneys and other tissues will use ketone bodies pretty readily. Then lastly is number 9, just on the left here, and this is going to be the breakdown of muscle glycogen. So skeletal muscle cells can break down glycogen into glucose 6-phosphate, which will undergo glycolysis and can provide ATP for muscular contraction. The term fasting means going without food for many hours or even a few days. I have that term down here. Whereas starvation, this implies weeks or even months of food deprivation or even just inadequate food intake. Now, people can survive without food for 2 months or more if they drink enough water to prevent dehydration. The most dramatic metabolic change that occurs with fasting and starvation is the increase in the formation of ketone bodies by hepatocytes. During fasting, small amounts of glucose will undergo glycolysis to pyruvic acid, which in turn can be converted to oxaloacetic acid, and acetyl-CoA can enter the Krebs cycle by combining with oxaloacetic acid. Your body produces more or less heat depending on the rates of its metabolic reactions. Because homeostasis of body temperature can be maintained only if the rate of heat loss from the body equals the rate of heat production by metabolism, it's important to understand the ways in which heat can be lost, gained, or conserved. So the term heat, this is a form of energy that can be measured as temperature, and it's expressed in units called calories. So heat is measured as temperature, and it's expressed in calories. Sometimes just CAL. So a calorie is defined as the amount of heat required to raise the temperature of 1 gram of water by 1 degree Celsius. So that's going to be our definition for a calorie. Amount of heat required to raise the temperature of 1 gram of water by 1 degree Celsius. So that's what a calorie is. The overall rate at which metabolic reactions use energy is called the metabolic rate. So as we've kind of already learned, some of the energy used to produce ATP, and some of this energy is used, or it's going to be released as heat. Because many factors affect metabolic rate, it's usually measured under standard conditions, and these are going to be where the body is in a quiet, resting, and fasting state. So these are going to be the standard conditions that we usually measure metabolic rate at. The measurement that's obtained under these conditions is called our basal metabolic rate, BMR. The production of body heat is proportional to metabolic rate. And several factors affect the metabolic rate and therefore affect the rate of heat production. Our first one being exercise. So during strenuous exercise, the metabolic rate may increase to as much as 15 times the basal rate. In well-trained athletes, though, the rate may increase up to 20 times. The next factor that can affect metabolic rate are hormones. So the thyroid hormones are actually the main regulators of BMR. BMR will increase as the blood levels of thyroid hormones increase. The nervous system is another factor that affects our metabolic rate. So during exercise or in a stressful situation, the sympathetic division of the autonomic nervous system is stimulated, releasing epinephrine and norepinephrine, which will both increase the metabolic rate of cells. So we'll just put increase, not our basal metabolic rate, but increase our metabolic rate, MR. Body temperature will also affect metabolic rate and therefore heat production. So the higher the body temperature, the higher the metabolic rate. Ingestion of food also will raise the metabolic rate 10 to 20% due to the energy costs of digesting, absorbing, and storing nutrients. Age is also a factor. So the metabolic rate of a child in relation to its size is almost double that of an elderly person, and this is due to the really high rates of reactions that are related to growth in children. And then we have a couple of other factors that also do affect metabolic rate. These are things like gender. Metabolic rate is generally lower in females than males, except during pregnancy and lactation. Climate is also another factor. Generally, metabolic rate is lower in tropical regions than in other regions. Sleeping as well. Your metabolic rate is decreased or lower when you're sleeping as opposed to when you are awake and active. And also malnutrition. So you're going to have a lower metabolic rate if you're malnourished. Now we will just quickly go over methods of heat transfer. Maintaining normal body temperature depends on the ability to lose heat to the environment at the same rate that it's produced by metabolic reactions inside your body. Now there are 4 ways that heat can be transferred from the body to its surroundings. These are by conduction, convection, radiation, and evaporation. So the first one being conduction. This is the heat exchange that occurs between molecules of 2 materials that are in direct contact with each other. So heat exchange by direct contact. An example of conduction would be losing body heat to a solid object, like a chair, clothing, or jewelry. You can also gain heat by conduction as well. For example, by sitting on a hot rock in summer, going to increase your body temperature by conduction that way. Convection is our next method of heat transfer. Now, this is the transfer of heat by the movement of a fluid, a gas or a liquid, between areas of different temperature. So contact with air or water with your body results in heat transfer both by conduction, because the molecules of air and water are directly contacting the molecules of your body, but also by convection, because it's going to be that fluid movement over your body. Then we have radiation. This is the transfer of heat in the form of infrared rays between a warmer object and a cooler one without physical contact. So pretty much everything emits some type of infrared rays, and your body loses heat by radiating more infrared rays or infrared waves than it absorbs from cooler objects. And then lastly is evaporation. So this is the conversion of a liquid to a vapor. And then all the heat that was in that liquid goes with it when it leaves the surface of the solid that it's on. So under typical resting conditions, about 22% of heat loss occurs through evaporation of water from your skin surface. The control center that functions as the body's thermostat is a group of neurons in the anterior part of the hypothalamus, the preoptic area. Now, this area receives impulses from thermoreceptors in the skin and mucous membranes, as well as the thalamus, or the hypothalamus rather. Nerve impulses from the preoptic area will propagate to two other parts of the hypothalamus. These are known as the heat-losing center and the heat-promoting center. When these areas are stimulated by the preoptic area, this sets into operation a series of responses that will lower body temperature or raise body temperature respectively. So here we have a negative feedback response for body temperature. So if the core body temperature declines, mechanisms that conserve heat and increase heat production are going to act by these negative feedback loops. So thermoreceptors in the skin and hypothalamus will detect this decrease in core body temperature and will send nerve impulses to the preoptic area as well as the heat promoting center and neurosecretory cells in the hypothalamus, as well as the thyrotrophs in the anterior pituitary. In response, each effect responds in a way that helps to increase core temperature to a normal value. So we're going to get vasoconstriction in the skin to decrease heat loss through the skin. The adrenal medulla will also release hormones to increase cellular metabolism, increase body heat that way. The skeletal muscles will contract very quickly, what we call shivering, also increases body temperature. And then the thyroid gland releases its thyroid hormones, which will also increase metabolic rate. So all of these effects having some type of effect to all work together to increase body temperature and bring us back to homeostasis. Most mature animals and many men and women maintain energy homeostasis. So this is the precise matching of energy intake in food and matching it to energy expenditure over time. So energy homeostasis is going to be matching, so this is matching energy intake and the amount of food you eat with energy expenditure. Now, when the energy content of food balances the energy used by all cells of the body, this is where body weight remains constant, unless there's a gain or a loss of water, but generally energy intake, if it balances out with energy expenditure, the body weight will stay the same. So energy intake, this is really going to depend only on the amount of food consumed and absorbed. But there are three components that contribute to total energy expenditure. So this is based on your basal metabolic rate. This counts for about 60% of energy expenditure. Physical activity will add another 30 to 35%, but this percentage can be lower in sedentary people. And then lastly is food-induced thermogenesis. And this is going to be the heat produced while food is being digested, absorbed, and stored. This represents about 5 to 10% of the total energy expenditure. Within the hypothalamus are clusters of neurons that play key roles in regulating food intake. Two hypothalamic areas involved in the regulation of food intake are the arcuate nucleus and the paraventricular nucleus. Now there is a gene that we found in mice, and it's actually called obese, or I guess they named this gene obese. And this gene causes overeating and severe obesity in its mutated form. The product of this gene, the protein that it codes for in its non-mutated form, is the hormone leptin. So obese codes for leptin. Because obese is the gene, leptin is the protein. In both mice and humans, leptin helps to decrease the amount of adipose tissue and the total body mass. So it helps to decrease that total adiposity. When leptin and insulin levels are low, though, neurons that extend from the arcuate nucleus to the paraventricular nucleus release a neurotransmitter called neuropeptide Y. And this will stimulate food intake. Other neurons that extend between the arcuate and paraventricular nuclei release a neurotransmitter called melanocortin. And this is kind of similar to the melanocyte stimulating hormone, MSH. But melanocortin will actually inhibit food intake. So leptin, stimulated with the release of melanocortin, will act to inhibit food intake. Nutrients are chemical substances in food that your body uses for growth, maintenance, and repair. The 6 main types of nutrients are water, carbohydrates, lipids, proteins, minerals, and vitamins. Obviously, we know water is needed in the largest amount, about 2 to 3 liters per day. Different populations around the world eat really radically different diets that are adapted to their particular lifestyles. However, many experts recommend the following distribution of calories. About 50 to 60% from carbohydrates, with less than 15% from simple sugars, less than 30% from fats. Triglycerides are going to be that main type of dietary fat, with no more than 10% coming from saturated fats, and then about 12 to 15% from proteins. Minerals are inorganic elements, so they are compounds without carbon, and minerals occur naturally in the Earth's crust. In the body, they appear in combination with one another. And minerals are actually really important to maintaining a healthy body. Organic nutrients required in small amounts to maintain growth and normal metabolism are called vitamins. Now, unlike carbohydrates, lipids, or proteins, vitamins don't provide energy or serve as the body's building materials, but most vitamins with known functions they act as coenzymes. So they're still very much needed, but they don't act as building materials. Calcium and phosphorus form part of the matrix of bone. A major role of minerals is to regulate enzymatic reactions. So calcium and iron, magnesium, and even manganese, which I think is on the next slide, but these are all going to be minerals that constitute for some coenzymes. Magnesium also serves as a catalyst for the conversion of ADP to ATP. And minerals like sodium and phosphorus work in buffer systems, which help to control the pH of body fluids. Sodium also helps to regulate the osmosis of water, and along with other ions, sodium is going to be involved in the generation of nerve impulses. So just some of the minerals that are really vital to the body here. So just kind of that second page of that table here, manganese is on this one, as well as copper and cobalt. Again, very important minerals that are vital to body functions. Now, most vitamins cannot be synthesized by the body, and they must be ingested in food. Other vitamins, like vitamin K, are produced by bacteria in the gastrointestinal tract, and then they are absorbed. Now, vitamins are divided into 2 main groups. We have fat-soluble, which is going to be on this slide, and then water-soluble. So the fat-soluble vitamins A, D, E, and K are absorbed along with other dietary lipids in the small intestine and they're going to be packaged by chylomicrons. So those are going to be those fat-soluble vitamins packaged in chylomicrons. So they cannot be absorbed in adequate quantity unless they are ingested with other lipids. Fat-soluble vitamins, they may be stored in cells, particularly in hepatocytes they can be stored in. Here we have the water-soluble vitamins. This includes several B vitamins and vitamin C, which is actually going to be on the next slide, but these are going to be dissolved in body fluids. Excess quantities of water-soluble vitamins are not able to be stored, but instead, when you take excess water-soluble vitamins, they are excreted in the urine. So here we have vitamin C, biotin, the rest of the B vitamins as well, and folic acid. So again, when you take like a lot of excess vitamin C, say you take 4 vitamin C chewable tablets, for example, your body's only going to take what it needs, and then the rest of that, the excess, is not able to be stored, so you're just going to excrete it in the urine. And that is the end of chapter 24.